So in that study, we actually needed an HLA-A1 restricted EBV-specific T cell population. So that was our starting point for the project that I discussed a few minutes ago. Because we need to make this risk analysis for both the CMV, EBV, and the other virus, but also with every HLA restriction. And one HLA-restricted population that we did not have in our lab was HLA-A1 restricted EBV-specific T cell populations. And when we first started out, we tried to isolate these T cells. It was actually very difficult. So we looked back into literature, and there were so many reports mentioning that there are no HLA-A1 restricted EBV-specific populations described. And what was more interesting is that patients that were HLA-A1 positive were more prone to develop EBV-associated lymphomas, but also nasopharyngeal carcinomas and gastric carcinomas. So there was something wrong with this HLA molecule in these patients, that they somehow could not make a good, adequate EBV-specific response. So we developed a few different tools, peptide MHC tetramers, based on a prediction algorithm that is just online, NetMHC, many people know it. And we made 6 different peptide MHC molecules that were restricted to this HLA-A1 molecule. And with one of them, we were able to successfully isolate HLA-A1 restricted EBV-specific T cells. And they recognized the LMP2 antigen and the epitope ESE. And what is nice about this LMP2 antigen is that we know that it's expressed in certain EBV-associated type 2 or type 3 malignancies. Some DLBCLs express EBV in a latent form that expresses LMP2, but also nasopharyngeal carcinomas and some other lymphomas too. So we thought we could perhaps use this population for in future treatment of these patients that have no other treatment option left. So this was all done in vitro. So we tested this T cell population in vitro. We isolated the T cell receptor from this population, and we CRISPR-Cas the endogenous T cell receptor in primary cell from a third donor out, and then we introduced our TCR, which was specific for this LMP2 restricted to HLA-A1. And we demonstrated that that these T cells that when transduced with this EBV LMP2 specific TCR were shown to specifically recognize and also kill LMP2 expressing malignant cell lines. So we had 3 different malignant cell lines, and we also tested them against EBV LCLs, which is basically a B cell that has been transformed by the EBV virus and thus also naturally expresses this LMP2 protein, and they were all successfully lysed and killed. So this could be have important value for in future T cell receptor gene therapies, or but we have not continued with this in any in vivo model, since there is no really good in vivo model to describe this. So that's a bit of a shame, but maybe in the future we could do, we aim to start a clinical trial of this TCR.